Hi everyone, our study is the analysis of self-targeting spacers reveal functional links between CRISPR systems, aminoacyl-tRNA synthetases, and the prokaryotic membrane. So CRISPR systems are genetic systems that allow bacteria and archaea to defend from mobile genetic elements such as bacteriophages by inserting small pieces of the phage's DNA on its own genome. These pieces are called spacers, and the sequences they come from are referred to as protospacers. Although many spacers come from phages as expected by the immune function of CRISPR systems, the presence of self-targeting spacers is widespread in prokaryotes. However, its functional role is still unclear. Some researchers have proposed that its presence is an error that causes autoimmunity, while others suggest it could be an adaptation for other roles of CRISPR systems not related to immunity. In this study, we analyzed self-targeting spacers of CRISPR systems and characterized its protospacers, finding between them a high presence of membrane proteins, aminoacyl-tRNA synthetases, and ATP-binding proteins. This is a novel report that supports other research linking CRISPR systems to membrane proteins and could explain the reported relationship between antibiotic resistance and presence of CRISPR systems in prokaryotes. So in this study we analyzed the CRISPR-Minor self-targeting database, which is based on the studies of Rauch et al., Stern et al., and Waters et al. This database consisted on a list of self-targeting spacers with its genomic coordinates along with those of its corresponding protospacer. However, a characterization of the nature and function of those protospacers was lacking, as well as the characterization of the CRISPR systems themselves. In order to characterize protospacers and CRISPR systems, we obtained information available in the NCBI by using BioPython and analyzed the information with the Pandas library. In our scan against the ProCyte database, we found 91 hits in 71 sequences. Of these hits, 44 corresponded to a prokaryotic membrane lipoprotein lipid attachment site profile, indicating a close association between self-targeting spacers and membrane proteins. It is remarkable to note that many CRISPR-associated proteins were also found as protospacers, for example, long proteases, indicating a functional link that remains to be discovered or a possible autoregulatory mechanism. Another interesting aspect is the recurrent association between CRISPR-Cas and aminoacyl-tRNA ligases. In our study, alanine-tRNA ligase and leucine-tRNA ligase were the most targeted aminoacyl-tRNA synthetases, which could be explained by the fact that these are common amino acids in proteins, and thus targeting these enzymes could be an efficient way to shut down protein synthesis when bacteria are infected with a virus and the CRISPR-Cas system is active. Recently, Jane et al. Have shown that Leptotrichia shahi Cas13a is able to cleave anticodons of certain transfer RNA, supporting this hypothesis. Previous analysis of CRISPR-Cas systems and CRISPR-associated proteins has revealed a mysterious connection between CRISPR-Cas and membrane proteins, and our results support this association. The regulation of membrane proteins by CRISPR-Cas could assist different functions. For example, it could be ligated to immune evasion in pathogenic bacteria, as has been shown in Campylobacter jejuni and Francisella novicida. Moreover, membrane proteins can also be necessary for phage infection. So one possibility is that some self-targeting spacers are directed against those proteins. In summary, this study highlights the importance of studying self-targeting spacers and supports the recent associations found between CRISPR-Cas systems and membrane components, an aspect of CRISPR-Cas biology that requires closer attention. It also highlights the fact that while non-canonical functions of CRISPR-Cas are not something new, they may be more common in prokaryotes than previously believed.